But I'd really like to kind of dig into as well. So one of the things you were talking about is you were, first of all, you wanted to return to your roots in the circular RNA space, right? You wanted to kind of continue the research in that with Thomas. But it also sounds like you saw a lot of benefits to sort of integrating it into the viral vector space, gene therapy space, right? So I'd be curious to understand a little bit more of your interest in jumping into the gene therapy space. Where were some of the biggest limitations that you were seeing? Where did you really find that there was a good window for you guys as a circular RNA company to start playing in the AAV, right, gene therapy world?
So, like I said, we started off with an oncology angle, and the company really by background is an immunotherapy company. And we wanted to leverage this adenoviral platform, and the initial idea was to express in tumors, so make cancer therapeutics leveraging the circRNA. And at that point, no one had moved in that direction yet.
Yeah.
So largely a virgin territory. So first of all, what you need is to build an expression construct that makes circular RNA. And of course, you start from linear DNA. So the viral genome is linear DNA, and you need to put the recipe inside of that DNA so that the RNA that is transcribed circularizes. So that's where we needed to start. And in nature, this is quite an inefficient process. I think it's been estimated that less in a natural circular RNA locus in a human cell, for example, only about 5% of the RNA will actually circularize. The other 95% gets degraded or have some other function. So in order to make sufficient circular RNA, it took quite substantial work to optimize the structure, design, sequences. And over time, we've improved this at least tenfold.
Okay.
So we built what we call CircVec. So this is a genetic cassette, double-stranded DNA, that you can put into anything that is a DNA or viral DNA format, and it will make circular RNA inside of a cell. So that's the fundamental technology that we developed. Now that's sort of independent of where you utilize it. So the story we first told was that we were going to use this when we went to investors and partners. Now we're going to use this circular RNA expression technology to make better oncolytic viruses, because we were an oncolytic virus company. But I think we underestimated the negativity of the oncolytic virus modality, and then people saw us as, oh, so you're not a circular RNA company, you're just a turbocharged oncolytic virus company. I thought still that would be pretty cool.
I mean, that's a great branding. We're turbocharged. I mean, come on.
But it was difficult to get anyone sufficiently enthusiastic about that idea, and then the more kind of data we generated and demonstrated expression advantage, longer durability compared to expressing from mRNA, higher expression level. I mean, we get about 10 times higher protein outputs simply by switching from linear mRNA to circular mRNA when expressing a reporter or whatever. And in mouse models, we see several months of longer duration.
Okay.
So once we start showing this data to people, they say, forget about cancer here. This you should deploy for gene therapy. So our pivot to gene therapy was to a large degree driven by this demand from companies and investors we spoke to, just telling us that, look, guys, this is something I would really like to see being deployed in the gene therapy setting. And of course, it makes sense. The biggest hurdle in gene therapy is, besides the delivery aspect, is getting sufficient protein expression for good enough durability.
Yeah.
So anything you could do to boost that expression is going to be an advantage. And I know Tom Barnes, the former CEO of Orna Therapeutics, you had on the podcast, and he said that he was convinced that everyone over time is going to switch from in vitro transcribed mRNA to circular RNA, LNP packaged circular RNA, because it's simpler to make, it's cheaper to make, it's more durable, it's going to make more protein. It's, at least so far, there is no evidence that should suggest that you would, once this is firmly established technology, why you would stay with mRNA. Everyone's going to switch. It seems obvious. And I think you can extend that to any virus or DNA-based therapeutic in the future. Like everyone will switch from linear mRNA to circular mRNA. Everyone is going to switch from mRNA expressing vectors to circular RNA expressing vectors because they will make more protein with better durability.
And hopefully reduce the dose too, right? Reduce the amount of virus you need.
Yes, exactly. And coming back to your initial question on the AAV, so one of the biggest caveats of AAV gene therapy today is that you need to give really high doses. To get sufficient protein expression, you need to give these massive viral loads that can drive up toxicity, can be lethal in some situations, increases manufacturing cost and complexity. Now, if you simply make the switch, where instead of expressing your genetic payload in the AAV from mRNA, is express it from circular RNA, and that helps you boost the protein output, that would be a massive advantage. Now, and our data suggests that we can get to 10, 20, maybe 100-fold better protein outputs from an AAV. If you can reduce the dose of AAV gene therapy by 10-fold, that would be a big thing. That would mean lower cost, more accessible to patients, and suddenly you can treat more diseases.
Yeah.
So you heard it here first. You may soon be editorial and community director of Advancing Circular RNA, because that will be the way. This is the way.
Many flavors, Matt.
All right. I want to get too wrapped up in the hype. I want to back up just a little bit though. You made an interesting point. You said you said you'll do this on two conditions. One, circular RNA is going to be the focus, and two, you bring Thomas along with you. And this was pre-data. We know that data drives confidence in the investor community, in the scientific community, in your own internal community. But when you made the decision to make this change, this circle or pivot, however you want to look at it, yeah. It was pre-data. So I'm curious, like from a business leadership standpoint, what other resistance you may have faced and had to work through prior to having that data that kind of generated the direction, right? That created the impetus for like, okay, well, this is where we're going to go with this. What other resistance did you have to overcome, and how did you overcome it?
So the parent company of that initially owns Targovax and owns these immunotherapy programs is listed on the Oslo Stock Exchange in Norway. That was listed in 2016. So it had been on the stock exchange for a long time. And of course, there were many tired investors because they had invested in clinical stage programs, and these had kind of, I wouldn't say failed data-wise, but not sort of materialized into later stage development, and the share price had fallen. And there's not a lot of sophisticated biotech investors in Norway. It's hard enough to understand a clinical stage program, but when we come then, suddenly now we're a company working on an early RNA platform that generated, I think, a disconnect with the shareholders we had at the time. Mm-hmm. And also when you're listed, I think most listed companies, at least when you've been listed for a while, you really need to be in the clinic that's the right time of development. So it's not ideal to be a early preclinical stage company and listed. So we were kind of grappling with how to square this circle where the investors we have are not really that interested in investing into this type of early stage technology or platform, and then that's much more attractive to VCs to come in early. Seed investors and VCs, that's what we're really after, but they don't buy listed shares.
Yeah.
So from the get-go, when we set this up, we built it as a privately held subsidiary, and for a variety of reasons, we set that up in Sweden via a lab at the Karolinska Institute. So we already from the start incorporated this as a standalone entity within the group. It's still fully owned by the parent company, and then you could view the listed parent company as the seed investor. So the listed parent provided the funding to set the company up, generate that first data, because as you rightly point out, it's difficult to fundraise on a slide deck. It was maybe doable 2020, 2021 for a little while.
It was. Not so much now.
Now the reality is back. you need to have a reasonable data package before you can do a solid financing. Yeah. So we've slowly built this from scratch, and by doing it from the get-go, we have all the employees, the IP, the data, everything is contained in this subsidiary in a different jurisdiction. So I think we're kind of building this as a private company, which we will eventually finance and spin out when the data package is mature enough, when the parent is no longer sort of suitable or capable of funding it to its full potential. I think that's time to let that ship loose from the group, and then the group holding company continues to run this KRAS cancer vaccine clinical development.
Did you turn to the West? You mentioned that in Oslo and in Norway, the appetite for investing in that early stage stuff is a little bit less than perhaps in Europe and the United States. Did you sort of expand your horizons on that front as well?
Yeah, we've been doing extensive roadshows and talking to investors. I would say being a smaller Nordic biotech these days, it's easier to get traction with the European VCs.
True. Mm-hmm.
I have a sense that there are fewer deals happening these days. And many investors, I think, are overloaded with opportunities. So I get the sense that the US VCs have more than enough with local opportunities. It's more challenging to get attention as when you come from sort of a small, weird country. When there is plenty of US, they don't even have time to review all the opportunities that are available. So it's certainly been trickier the last, I think since the COVID bubble burst. It's been harder for the Nordic or the European companies to get a lot of meetings and interest in the US. I'm not saying it's zero, but it's less than it was before. Whereas in Europe, I sense the opposite. Now you tend to see higher valuations in the US. I think many European VCs are to some degree preferring local investments because they see it as an opportunity to get better valuations.
Mm-hmm. Are you finding... Sorry, go ahead.
Yeah, so all the investors we speak to, I'd say about 70-80% are European.
Mm-hmm. Okay. I'm curious, when you were starting to fundraise for Circio, have you seen that there's been shifts? Sort of, obviously there was a time when you could get as much money as you wanted with as little data as you had, right? And right now you guys are in the process of getting as much data as you possibly can, but are you finding that you're facing similar pushback? Is there from investors as you're striving to continue to raise more money? Where is some of that resistance, do you think, coming from? What are some of the biggest challenges you're facing in engaging the investor community today, particularly as you're working with circular RNA and potential viral delivery, right?
So I think circular RNA is definitely super hot at the moment. Mm-hmm. It's as compared to when I've been trying to shop around a cancer vaccine and an oncolytic virus in the past, it is more difficult to people don't automatically open their doors. It's not something they have high on their shopping list. So you need to come with something extremely compelling. Mm-hmm. When you come with a circular RNA program, everyone wants to listen. This is just something everyone is really curious about these days. Right. I would say the biggest difference from a couple of years back to now and challenge to some degree is it's much harder to sell a platform now or to get investments into a platform. And that's in its essence that what we're developing. Our tool is CircVec, circular RNA expression is a genetic cassette that you could put anywhere. It can be used to whatever you want to express from a DNA or a virus or even in a VLP context. So it has a lot of versatility, and I don't think we've come to the point where we reached its maximum potential. Mm-hmm. And we still think it can get more potent than it is. And at the same time, we're still testing where does it work, which vectors are the best, what tissues are the best. So this we still need to figure out. But investors these days want a therapeutic candidate. You need to have a lead program. Sort of, it's coming with a platform play is difficult to sort of get money in the bag for. So we need to have two thoughts in our head because it's crucial to keep expanding, building, understanding, developing the platform. but at the same time, having a therapeutic lead spearhead, which clearly sort of says this is the direction we're going. This is how we're going to get to a clinical proof of concept.
So what are you doing to try to confront that? You know, the lack of interest in a platform company, right? Have you had to change your strategy at all?
And when we started this, it's difficult because we came from cancer. Now people tell us you need to go do, several investors said, when you're doing gene therapy, come back. Okay, so we have to do gene therapy. How can we deploy this technology in gene therapy? Well, AAV is obvious because AAV has these caveats. I think it's the lowest technological risk. Okay. It already works, clinically validated, manufacturing is understood, it's approved for several diseases, and it has a lot of caveats. So we can improve on this. So we said, let's see if we can simply just improve on existing AAV technology. You take AAV, express a payload via circRNA instead of mRNA. That's the only difference. Everything else can be the same. So you could then develop something, just go head-to-head against an existing gene therapy. Take an AAV gene therapy and switch to our expression system, reduce the dose. If you could do that and show just in a non-inferiority trial, I think that's something that could be approvable. So this is where we started out. Then the question is, what is the best disease to pick? And do you either go that direction where you try and go head-to-head against something that's already existing and reduce the dose, or do you pick something where AAVs weren't sufficiently effective in the past, and then switching to a circRNA could kind of get you over the threshold to make it clinically relevant?
Right.
So that's something we debated back and forth, and I think both options are still in play. But I think in the end, we opted for the latter because once you start to go head-to-head against something else, you need to do randomized trials. In gene therapy, it's often diseases with very few patients available. So we prefer to go to something that is still kind of virgin territory. So we decided, okay, let's pick a disease where AAVs have been tried but didn't make enough protein. So we can boost the expression level and kind of have a successful therapeutic product that way. And from there on, we were all oncologists mainly. The cancer is the area we knew, so we had to sit down and learn about genetic disease. So we screened thousands of genetic diseases you'd never heard of.
Great study parties.
We had a very talented analyst at the time. It was excellent compiling this information in true McKinsey style. It's a perfect McKinsey project, right? Coming back to that. Quickly sort of making 5-pagers for each disease and have a good heat map where you can score what is the better and worse options. And then in the end, we settled on a short list of disease targets. Mm-hmm. All the while working on the platform in the background.
Mm-hmm. Mm-hmm. Good.
Do you want to?
Matt and I are both like jumping for the microphone.
We're like arm wrestling. I want to talk about the business and
I'm like, give me the science. All right, I will allow you to.
Thank you. I appreciate that.
Host your podcast, Matt. Yeah.
I think we've done a nice job covering sort of the investor challenge perspective that Circio faced. Now I'm curious about organizationally and particularly from a personnel level, making this shift. I mean, you mentioned the study part. I envision like all these oncologists. Anyone who's watching the podcast right now on the website can see that Erik is in his summer home near a fjord in northern Norway. Is that where
the south coast of Norway, close to Kristiansand, if you know, like the southern tip of Norway.
Complete opposite.
So I'm picturing him having like these all these oncologists gathering at the summer home in the fjord.
It's at night though, and there's like candles everywhere.
Candles, yeah. There's a lot of writing going.
Maybe a giant PowerPoint.
Yeah.
How really, yeah.
So that there was that, right? Like you had to bring that.
Painted the scene somewhat in a Renaissance fashion. Style of Da Vinci.